Hi, everyone. I should just preface by saying that like most of my work is like wet lab, so I'm actually new to composition biology. But we're just trying to do this large scale transcriptomic study. So the study is basically the transcriptomic profiling of cell adhesion molecules, specifically synaptic adhesion molecules in a developing cerebellum. Yeah, so the question is basically, imagine you have a field populated by many neurons, and a neuron enters this field. How does the neuron know which receiving neurons to synapse onto? And so the hypothesis in the field is that cell adhesion molecules mediate the specificity of this interaction, where like this one receiving neuron called a dendrite might express this cell adhesion molecule, another one might express a different one, and another a different one. So if the incoming neuron expresses this magenta cell adhesion molecule, it will recognize the same cell adhesion molecule on one of the receiving neurons, and that will specify the synaptic connection. And so the synapse will be formed. And so to study this question of synaptic adhesion or synapse specificity, we turn to the cerebellum, the brain region involved in balance, motor coordination, and other cognitive functions. And specifically, we look at the cerebellar cortex. And so this is what the cerebellar cortex looks like. So starting from the bottom, this is where information comes in through the mossy fiber. And the mossy fiber synapses onto what are known as granule cells. These are the only excitatory cells in the cerebellar cortex. And so these granule cells will pass information upwards and excite all of these cells up here, but mainly the Purkinje cells, which are the only outputting neuron of the cerebellar cortex. And so the layers of the cerebellar cortex are the granule layer at the bottom, Purkinje layer, and then the molecular layer. And each of these layers have what are known as interneurons, which are just inhibitory cells that dampen the signal of each of these cells. So in the granule layer, you have Golgi cells, which are the granule layer interneurons, GLI. And in the Purkinje layer, you have Purkinje layer interneurons. And in the molecular layer, you have basket cells, which synapse onto the cell bodies of the Purkinje cells, and stellate cells, which synapse onto the dendrites of the Purkinje cells. And together, they make the molecular layer interneurons. And so the field is basically wanting to find markers firstly, and then find markers of adhesion molecules. And so in 2021, an RNA, single nuke RNA-seq paper was published where they isolated many cells of the cerebellum, but this was in the adults. And so this paper generated many markers of supporting cells of the cerebellar cortex, but also for granule cells, Purkinje cells, and all of the interneurons, which are the focus of our study. But our question is, what are the transcriptional profiles of the connectivity molecules in these interneurons, and how does this develop over time? And so to study this question of development, we have to turn to a developmental RNA-seq data set where we've selected cells all the way from embryonic day 18 all the way to P16, so just postnatal day 16. And so my goals are firstly to identify the cell types in this embryonic data set, and then to profile adhesion molecules. So the first thing I did was I identified granule cells based on a marker that's expressed in the adults. And granule cells are the most numerous cell in the brain. And so to simplify the UMAP, I removed them. And then on the simplified UMAP, I identified the expression of ASCO1, which is the transcription factor expressed by interneurons. And so those would be my developing interneuron population. And then TFAP2B, which is a transcription factor of interneurons all the way from development to adulthood. And so these would be the precursors and then the developing interneurons from E18 to P16. But to find the markers of each of the actual interneuron subtypes, we'd have to use the adult set because that's where the markers are most confident. So then what I did was I combined the P60 adult data set to the developing data set. And I should also note that so this data set has MLI1, so molecular layer 1, 2, then granule layer interneuron, and then Purkinje layer interneuron. But I should note that the molecular layer interneurons were separated into two subpopulations in the adult RNA-seq data set as MLI 1.1 and 1.2. And these are the cells that we'll focus on. So I should remind you that the MLIs are the stellate cells and the basket cells. So when I didn't realize that the resolution would be so low, but this is basically just showing the cell types. So this is what happens when I add the developmental interneurons to the adult interneurons. And so this is just showing the time. So you can see that all the P60 cells have been added onto the developing cells, and that for some of them, they've actually split into the two. This is MLI1 and that's MLI2. And so I'll just focus on the MLI population. And so this slide is to show you that basket cells and stellate cells are not separated by MLI1 and 2, but in fact, they're all included within MLI1. And so here at the bottom are basket cells synapsing onto the cell bodies of Purkinje cells, and at the top are stellate cells that synapse onto the dendrites of Purkinje cells. And so MLI1 is identified with this marker SORX3. And this is really faint on the screen, but you can see that this is single molecule fluorescence in situ hybridization. So you can see that SORX3 labels cells all the way at the top here, which is the region of the stellate cells, and cells at the bottom here, which is the region of basket cells, indicating that SORX3 might be positive in both basket and stellate cells. And then when we look at NXPH1, it's really faint, but in blue, there are cells that are sort of interspersed in this area, but they're not specific to either these stellate cells at the top or basket cells at the bottom. So this question was studied in depth by a previous student in our lab, Wendy Wang. And what she did was she identified the SORX3 population and separated it based on these two markers, CACNA1E and GRIM8, which you can see separate very well in this UMAP. And so first looking at the stellate cells, she showed that the stellate cells express SORX3, but they don't express the MLI2 marker, NXPH1. And then the basket cells also express SORX3, the MLI1 marker, but they don't express the MLI2 marker, NXPH1. But then when she looked at the expression of CACNA1E and GRIM8, she showed that the stellate cells have high expression of CACNA1, indicating that CACNA1 might be a good marker for these stellate cells. But then the basket cells didn't express CACNA1, instead they expressed GRIM8, which indicates that GRIM8 might be a good marker for these basket cells, all of which are included within the SORX3 population, which is MLI1. And when we look at the MLI2 population that are positive for NXPH1, she showed that they express NXPH1, but they don't express SORX3, and they also don't express GRIM8 or CACNA1E, suggesting that they're a completely different population, which she described as having properties that resemble short stellate cells. And so for this, just this preliminary description, we'll focus on MLI1 to capture the well-studied basket and stellate cells. So the first thing I did was I identified MLI2 from development all the way to adulthood. And also I should note that this part of this developmental cluster is positive for IGF-BP5, which is actually a Golgi cell precursor. So we'll remove both this and that, focusing just on this section of development and then MLI 1.1 and 1.2. And so I subsetted the data set, mapped it, and you can see that we have what can be a trajectory from development to basket cells and then to stellate cells, where the basket cells express GRIM8 and stellate cells express CACNA1E. And so the goal here is to profile cell adhesion molecules in both the stellate cells and the basket cells. So this is just a commonly studied cell adhesion molecule in the brain, cadherin 2, otherwise known as neural cadherin. It should be expressed by all neurons, and here you can see that it's expressed pretty equivalently in both basket and stellate cells. But just an example of our profiling goal, here is cadherin 4, which is very clearly expressed in basket cells, but not as highly in stellate cells. And then the synaptic adhesion molecule 1, CADM1, which is expressed very clearly in stellate cells, but not in basket cells. And so this is sort of the thing that we want to do, but in scale. So then our goal is, this is a summary, that stellate cells have CADM1 and basket cells have cadherin 4. The implications of this are that these molecules could be what's differentiating the type of synapses that are occurring on the dendrites of Purkinje cells versus their cell bodies. So this is sort of the thing that we want to do by generating an atlas of these cell adhesion molecules to allow for the generation of these types of hypotheses. And so just to summarize, our goal is to profile the expression patterns of these connectivity molecules like adhesion molecules. And we want to use both spatial transcriptomics as well as dissociated cells, where with spatial transcriptomics, we can leverage the segmented structure of the cerebellum because we know where each cell is. And so that can help us in our confidence. And also in early development, all the cells are sort of aggregated together. So then we can infer relationships based on their proximity. And we also want to use different developmental time points to capture changes in the dynamic expression of these molecules. And so I'm open to tips on how to, one, analyze this and how to display it. Because the challenge is that these cell adhesion molecules are not going to be expressed in isolation. It's likely that they'll be combinatorial. Like one neuron might have like 5 of these specific to it, and the other might have like 10. And we also want to see how this changes over time. So I'm open to hearing tips on that. So for the analysis of cell adhesion molecules, people typically use cell chat. And then for trajectory, people tend to use pseudotime. But I'm curious of how these two can be combined together. So that's pretty much all I have. So I just want to thank the FAVE Lab and Wendy Wang for doing most of the preliminary work, and my committee and collaborators. And thank you all for listening.